Hi everyone, thank you for inviting me, and I'd like to thank the other speakers for already having explained, giving the introduction about dynamic FC, so I can go straight to the point of my talk. So I will talk about the switching choreography of the connectome and how fast can we detect meaningful functional networks from fMRI data. So when I mean fast, is what is the fastest time scale we can go through, and meaningful is important, so I will focus the second part of my talk about looking at the meaning of these functional connectivity patterns that we find. So, okay, they already introduced this, but the dynamic nature of the functional connectome is the fact that if we use a sliding window, we see that there are different patterns of functional connectivity that we can observe in the functional connectivity matrix. But importantly, when we really look at the bold signal, it seems that there is the global fluctuation of the whole signal. Here it's 90 areas and they were smoothed below 0.1 hertz. But we see that from time to time, it seems that the bold signal from some brain areas disengages from the global signal. And whenever the window captures these epochs of this disengagement, we find these specific patterns of connectivity. So this gives, when we have the matrix, the time versus time matrix of FC dynamics, so we try to correlate the matrices of functional connectivity over time, we see these large squares that are very sensitive to the window size that we use. So if we use, here is 60 seconds, but if we would use 100 seconds, these squares would be proportional to that window. So we decided to try this measure. This was actually, so instantaneous bold phase synchronization was actually introduced by Enrico Glerian in 2012. And it consists in computing the bold phase over time using the Hilbert transform. So this is quite straightforward. There's a MATLAB function called Hilbert, and you just do angle Hilbert of the signal. And then you calculate the phase synchrony as the cosine of the difference of the phases, or 1 minus the sine. I think it's a very similar measure that was introduced. So when we do this, we can obtain at each instant of time an instantaneous functional connectivity matrix that captures the phase synchrony of the bold. So in more detail, whenever two brain areas are simultaneously increasing their metabolic demand, they have positive instantaneous IFC, and when they are simultaneously decreasing their metabolic demand, they also have positive, and they only have negative functional connectivity whenever the angle is bigger than 90 degrees. This is how the thing. So when we look at this matrix, okay, it's quite noisy. That's one of the problems of the instantaneous measures of dynamic FC. But if we look carefully, we see that there are some patterns that seem to appear and disappear over time, that look recurrent. But we want to see, is this meaningful? Does this matrix make sense? What are the networks that are coming out? Anyway, for a proof of concept, I just do the mean. So I collapse over time all these matrices, and I do the mean. And it correlates like above 99% with the static FC, the correlation matrix of the bold signals. So in a way, we are talking about the same thing. Doing the Hilbert phase and doing the phase synchrony, in the end, it gives us that static FC. But it allows us to extend this over time, to look at the temporal dimension of the matrix. But then again, if we do the time versus time matrix, okay, we got rid of the large red squares along the diagonal that were due to the window, but then the detection of recurrences is quite weak. So after a long time, we see that there is like a correlation of 0.3, 0.4 over time. So there's a weak detection of recurrences. So the new thing that I introduced here was to, instead of just looking at the instantaneous connectivity matrix over time, we look at the leading eigenvector of this instantaneous connectivity matrix. So the leading eigenvector is like the first principal component of that matrix. It's the one that captures the main connectivity pattern. So if we would want to divide the system into two communities, we could use just that vector. So in more detail, so here, for example, all the areas in red would be going in one direction, and the areas in blue, their bold signal is going in the more than 90 degrees in the opposite side. And that would be the pattern that is captured by the eigenvector. So in another, again, this is a simple function from MATLAB, Ix of the IFC one. And so you get a vector. And that vector has 90 values. And it has 90 values that correspond to 90 brain areas, because here we are using the AAL. And we can then, it has different signs, the areas, and it means that the areas that all have the same size can be assigned to one community, and the areas that have the opposite sign are assigned to another community. So this is based on the algorithm from Newman to make community detection. So I started using that algorithm a long time ago during my PhD, so it's a bit inspired on that. And okay, so again, to explain a bit what the eigenvector is capturing of the connectivity matrix. So if we have the high resolution matrix, and then if we decompose it into its principal components, here in this case, the first one represents 77%, then the second one, 22%, and the other ones appear almost unmeaningful. But importantly, I did this for all the studies I've been doing. The leading eigenvector mostly represents more than 50% of the signal. So in a way, you can represent the main matrix as a sum of the two principal patterns. And you see that, so we can represent the matrix that had, all the number of unique elements are all the elements above the diagonal in the first one, and the high resolution one. And we can represent it by just two vectors and their associated eigenvalues. So in a way, now we can plot simultaneously to the instantaneous functional connectivity matrix. We have a vector, the leading eigenvector, that we can plot on the cortical surface and have a look at what are the patterns that are matched with the. So I have to note that I had a problem with making them work at the same time, so they are not exactly representing the same pattern, but it's from the same data set and the same person. Anyway. So the idea is that we have the dominant connectivity pattern at each TR of the fMRI data set. So now, when we look at the time versus time matrix, we see that actually we can detect recurrences, so that go up to 0.8 after 200 seconds of scanning. So we can really see that the same dominant pattern, in a way, using the leading eigenvector is like getting rid of most of the noise, and we just focus on the main communities that are being formed. Okay, so then we find these recurrences, but are these recurrences, what are the patterns that are emerging? Do they make sense? Are they familiar functional networks? And are they similar across subjects? Can we compare them across subjects? So how to compare and detect recurrent states of functional connectivity? We need to use a classifier. So we can put all these leading eigenvectors that we had that represent the patterns from different subjects. We put them all together, and then we cluster them into a number of states. We can use k-means, but in the paper we also use HMM, and there's a possibility to use other measures. So the first part is instantaneous functional connectivity, leading eigenvector. And then here is the clustering part to look at the functional networks. So if we would be to represent all the patterns that the functional connectome is taking over time in this multidimensional space that here we represent in an eigenspace over its three principal components, this represents a cloud. This is a cloud of dots, and it's like the functional connectome is switching between this cloud in a continuous way. But when we apply a clustering, it will divide this into a number of, it will try to divide this cloud into a discrete number of states. Of course, we have to think, in general, this is a continuous space, but when we apply a clustering, we are dividing this into states. But what's important is, what are the difference between the cluster centroids of these states? And when we do, this one, when we look at the cluster centroids, when we divide it into 5 cluster centroids, we see that we can find one state that corresponds to the global signal, then there is one that corresponds to the default mode network, another that is the lateral frontal parietal network, another the somatosensory network, and then the visual network. and we look at the matrices. So the thing is, when we look at the matrices only, doesn't make much, it's hard to capture what it means, but when we project it into the cortical place, we start getting the meaning. But then we can divide this into 7 networks, and we have higher spatial resolution, we have more well-defined networks. We could go up to an infinity, as I said, because this is a continuous. So Marina did a great work. She's trying to, PhD student, she's trying to characterize, as we increase the number of states, how can we associate these to known functional networks? But the real number of networks that we want to divide depends also on the question being asked. Because if we are focusing on a very specific task or a very specific disease, maybe it's a functional network that appears a pattern very, very little in time, and then we need more. But in general, so then when we apply this thing, we not only identify the cluster centroids, but we can have the time course of these states. So for example, here, Caroline, she's applying this to a depression data set, and we can see like the blue states, we see when it occurs, we see how it looks like the pattern, and with this you can calculate the probability, which is the number of times it appears, the lifetime, so the number of consecutive epochs that that state dominates, and the switching profile. So it's the probability once you are in the blue state of switching to the green state or to the red state. So you can have a trajectory in this state space. So you can imagine this cloud, and we see how we are flowing through the cloud. So the first data set I applied this technique was to a data set of 100 healthy older adults aged more than 50 years old, and they were categorized according to their cognitive performance. They were the best and the worst among a cohort of 4,000 people who did high demanding cognitive tasks. And so in a way we wanted to see if performance, cognitive performance or task performance, like Rafael told, could be related to the expression of these different functional networks. So we found a significant difference that was at the beginning hard to interpret, but now I see other studies that are replicating a similar thing. So we find this state of global integration, or which is also the one with more variability, and it's the state that is less related to the structure, that increases in the people who are good performers. And it seems to be related with more awareness, with more selfness. And so here is the probability of this state, and the visual and the auditory networks, yes, were significantly reduced in the good performers. So when we compare this with the structural connectivity, we see that the states that were increased in the poor performers were the ones that related more to the structure. But this made us think, so there are reports saying that when we are asleep, also our functional connectivity is more similar to our structural connectome. So there was always this doubt whether these healthy elders that were poor performers, did they fall asleep? So we tried to correct for that. So it wasn't still exactly convincing. So then we had another amazing data set, and this was one of the most striking results we found so far, where the people were taking, it's an experiment with magic mushrooms. It's psilocybin. So we have a scan before the people were injected with psilocybin, right after they were injected, so they are scanned during the psychedelic state, and then they went back another week later, and they are scanned under a placebo. But this is a double-blind controlled thing. And so the thing was, we found that in all 3 non-psychedelic sessions, fMRI sessions, for example, the state 3, the fronto-lateral parietal network, was present 14% of the time on average across sessions. So 1 week later, it was the same probability. And during the psychedelic experience, this control network was totally disrupted. It didn't appear anymore. So this specific pattern. All the others, across 1 week, they appeared again. We could see again the visuals pattern, the somatosensory pattern, the DMN pattern. And in the psychedelic state, it seemed to move towards this global state of activation or correlated. It's probably the global signal here. So in a way, it seems that psilocybin is acting specifically in one functional network that is kind of disturbing the global signal, and so it's controlling the dynamics and maybe have a behavioral correlate. So again, going back to our cluster clouds, so this was before injection and after injection of psilocybin, and before the placebo and after the placebo. And here I want you to see the red cloud, which represents the frontoparietal network. You see that only after placebo injection, you really see that that cloud disappeared. So it shows here 7 networks, because this was the best division of the cloud into seven parts, so that we could exactly define the area that was removed under the psilocybin. So in a way, there's a molecule that acts in our brain by disrupting the probability of the functional connectome to go into that configuration. Sorry, my time is up, but okay, so this is another one in major depressive disorder. This is another state that is underrepresented. This is also with a high significant result. And it includes frontal, DMN, salience networks, including the insula, and the thalamus and striatum. So these are areas that are involved in emotional processing. And so we found it really also relevant for, so it's a kind of a behavioral correlate that we could find. Not only this, but so this significant network that appeared decreased in patients in remission from major depressive disorder, so they are at risk of becoming again with MDD. They also have significantly less probability of moving from that state to a prefrontal striatum that is also linked to pleasure, emotional regulation. So in a way, the fact that we are seeing how the functional networks are moving, how functional connectivity is evolving over time, is telling us what may be the mechanisms, the trajectories that are disrupted in these people that don't allow them to conceive the world or be so happy as the other people. So they are not able to access the pleasure system so well. This is another case where we see that the brain stimulation is restoring the repertoire of FC states with Parkinson's disease. This is also a significant effect. And finally, I'd like to discuss a bit, if you give me 30 seconds, yes, about what this is informing us in the sense of multi-stability. I like to see, so if we have these different connectivity patterns that drive us to do some behaviors or that are linked to some behavioral correlates, we can look at these patterns as attractor states that are shaped by our structural connectivity. Attractor states is a concept from dynamical systems theory, but I think we could apply it, and it could be a nice perspective to start looking at dynamic FC from this perspective. And in a way, this attractor landscape is modulated. So what we are finding is that it's modulated by certain drugs or certain neurotransmitters. So in a way, when the plot with the switching between networks is the one that shows, so in blue are the connections that are decreased under psilocybin. So if the frontoparietal, the top network, was a basin of attraction, it's like psilocybin is removing that basin of attraction in the dynamical landscape. And so the dynamics goes down to the other states. And so also we find this with DBS and cognitive training. And then I'd like to thank my network and funding. And I'd like to say that the codes are available on my GitHub, but the version is a bit outdated. So if you want to use, I can give technical assistance, drop me a line, an email, and we can maybe collaborate. Okay.
Thank you very much, Joana.